Namaste to all students. Kusha Vidya, myself J.V. and Dr. Kushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about different phases of cell cycle. So before starting about the different phases of the cell cycle, you have to know what is cell cycle. The cell cycle is a process in which a cell grows and divides to create a copy of itself. Means they can multiply the cell in number, and some organisms reproduce through the cell cycle. And in complex multicellular organism, the cell cycle is used to allow the organism to grow and to replace cell as they grow worn out. In animals, the whole cell cycle takes around 24 hours from start to finish. Some cells, such as skin cells, are constantly going through the cell cycle, while other cells are may dividing the rarely and if at all. Neurons, for example, don't grow and divide once they are mature. If we are talking about, you can understand the cell cycle phases by this diagrammatic representation. So here I am sharing the screen of slide by which you can easily understand phases of cell cycle. Here you can simply say this is a circular phases steps which you can easily understand. Cell cycle can be divided in majorly in four groups: G1 phase, S, G2 phase, and M phase. In G1 phase, this phase is basically having growth and normal metabolic rules, and this is first growth phase. Second is S phase. This phase basically required for the DNA replication. In this step, DNA multiply. And in the G2 phase, which is also required for the growth and preparation for the mitosis, this one is the second growth phase. And S phase is known as synthesis phase. And after completion, successful completion of the G2 phase, cell undergoes the mitotic phase, which is actually cell division phase. And this mitotic phase is having four major phases: prophase, metaphase, anaphase, and telophase. After the completion of this mitotic phase, cell can divide and can create their daughter cells. As I have already told you that cell cycle consists G1 phase, S phase, G2 phase, M phase. G1 phase is known as growth and synthesis phase. S phase is required for the DNA synthesis. G2 phase is required for the preparation for division. Cell division means they will prepare all the enzymes and proteins which are required for the cell division process. They prepare, and after the successful completion of this G2 phase, cell undergoes to the M phase, where cell start to divide, and finally they create two daughter cells. So M phase includes the overlapping processes of mitosis and cytokinesis. In the mitosis process, cell nucleus will be divided, and in cytokinesis process, cytoplasm will be divided. So after the completion of cytokinesis, cells will be divided completely by nucleus and by cytoplasm, and will create two daughter cells after the end of mitosis division and cytokinesis division. Now come to the detailed information about the gap phase 1 means G1 phase. It begins at the completion of mitosis and cytokinesis. Means this is the circular motion where when cell divide and can create the daughter cells, so they can undergo again in the cell cycle process. So G1 phase begins at the completion of mitosis and cytokinesis and lasts until the beginning of S phase. This phase is generally the longest of the four cell cycle phases and is quite variable in length. During this phase, the cell chooses either to replicate its deoxyribonucleic acid DNA or to exit the cell cycle and enter a quiescent stage, which is G0 phase. Means if any cell do not require further cell division. So they simply can go for the rest phase, which is G0 phase. And replication of the chromosome is restricted to one specific portion of interphase called S phase. One more thing, G1 phase, S phase, and G2 phase are collectively known as interphase. So, S phase from interphase is very much important, and it is restricted to only for replication process of chromosome. And this S phase is also known as DNA synthesis phase, which typically lasts about 6 hours. G1 phase was very longest phase, and for the S phase, this is 6 hours long. And in mammalian cell, the start of S phase, the actual initiation of DNA synthesis takes place several hours after the cell has committed to carrying out DNA synthesis. During S phase, each chromosome replicates exactly once to form a pair of physically linked sister chromatids. In animal cells, a pair of centrioles is also duplicated during S phase. The portion of interphase that follows S phase is called gap phase 2. Some cells can exit the cell cycle from G2 phase just as they can from the G1 phase. Combination of these three phases are known as interphase, as I have already told you. And M phase includes the overlapping process of mitosis and cytokinesis. Here you can understand M phase process in detail by this diagrammatic representation, where you can simply see all phases of M phase. This one is G2 phase. This is the cell which is having sister chromatids and chromosomes. They are loosely coiled replicated chromosome right now. They are having centromere and but one more thing, during the G1 phase, all enzymes which are required for the cell division process has been prepared during the G2 phase, and after a successful completion of the G2 phase, cell undergo into the mitosis division, and especially in the prophase stage, where cell membrane start to degrade, spindle fibers start to appear, chromosomes condense, and nucleolus start to disappear along with the nuclear membrane. Means nuclear membrane start to degrade or start to disappear, and nucleolus also disappears, and chromosomes start to condense, spindle fibers start to start to appear. After the successful completion of the prophase stage, cell undergo into the pro-metaphase stage, where you can simply see spindle fibers attached to the chromosomes and chromosome properly condensed. Now in this phase, next cell go into the metaphase stage, where chromosomes start to align on metaphase plate and they are attached to their spindle fibers. Now cell undergoes into the anaphase stage, where sister chromatids start to break, start to separate from each other. These are the centromeres. They have been divided, and after the division of centromere, these two sister chromatids have been separated from each other. Now these sister chromatids move towards their opposite poles, and after successful completion of anaphase, cell undergoes to the telophase stage, where again nuclear membrane start to reappear, nucleolus will start to reappear, and chromosome now decondense, and two nucleus, proper visible nucleus has been prepared at the end of the telophase stage. But in this phase, nucleus are divided. They have been created two daughter nuclei, but cell has not been separated and finalized daughter cells are not prepared till now. So cell undergoes from telophase to the cytokinesis process. In the cytokinesis process, cytoplasm divides. and parent cells becomes two daughter cells with identical genetic information. At the end of the mitosis process, you can simply see in mother cell, it was having two chromosomes and daughter cells are also having two chromosomes, means number of chromosomes remain same. There is no alteration, there is no change in chromosome number in daughter cells. So after the completion of mitosis process, two daughter cells prepared and chromosome number remains same as present in the mother cell or parental cell. So this is complete cell cycle where we have been discussed already that cell cycle majorly divided into M phase and interphase. Interphase again subdivided into three phases: G1 phase, S phase, and G2 phase. G1 phase is first growth phase and it is longest phase of the interphase. S phase is responsible for the DNA replication and also known as synthesis phase. And third phase of interphase is G2 phase, which is second growth phase and required for the preparation of enzymes and protein for mitosis phase. So after the successful completion of the G2 phase, cell undergoes to the mitotic phase, where it completes prophase, metaphase, anaphase, telophase, and cytokinesis phase. And after the completion of one cell cycle, one mother cell, two daughter cells will be created. So this is mitosis process. Now we are going to discuss about the cell cycle checkpoints, means cell cycle is working properly or not, if there is any error. So to check the regularity and to check the possibility of any error, cell naturally has decided some checkpoints. At these checkpoints, if any error can capture, so cell just completely arrest and cell do not go further for the cell division process. Cell arrest at that phase and stop to do cell division. So cell cycle checkpoints are also known as restriction points. These are the cell cycle control mechanisms in eukaryotic cells. These checkpoints verify whether the process at each phase of cell cycle have been accurately completed before progression into the next phase. Means if there is any error, so these checkpoints will check the regularity and the error. There are three main checkpoints that control the cell cycle in eukaryotic cells. They are G1 checkpoint, means G1 restriction point, second is G2 checkpoint, and third one is the metaphase checkpoint. First of all, we will discuss about the G1 checkpoint. This checkpoint is present at the end of G1 phase and before S phase. This checkpoint help in taking the decision of whether cell should divide, delay division, or enter a resting phase, means G0 phase. If there are unfavorable conditions for the cell division, means enzymes are not prepared, proteins are not prepared, and DNA has been damaged, or any other error, if they are just checking, then this restriction point restrict the progression to the next phase by passing the cell to G0 phase for an extended period of the time. Means they just forward that cell to the resting phase. This restriction point is mainly controlled by the action of CKI-P16, which is cyclin dependent kinase inhibitor P16. This restriction point or this checkpoint is controlled by this enzyme. The inhibited CDK do not bind with the cyclin D, which is one of the important enzyme required for cell division process. So when CDK inhibitor P16 inhibits the cyclin dependent kinase, so this inhibited cyclin dependent kinase do not bind to the cyclin D. When CDK and cyclin D1 are not bind to each other, so there is no further cell progression happens. Active cyclin D CDK complex is required for the phosphorylation of the retinoblastoma protein PRB in the nucleus. unphosphorylated retinoblastoma protein acts as an inhibitor of G1 by preventing E2F mediated transcription. Once retinoblastoma protein gets phosphorylated, E2F activates the transcription of cyclin E and E, which then interact with the CDK2 to allow for G1S phase in transition process. This brings the cell to the end of first checkpoint, which is unphosphorylated retinoblastoma inhibits the E2F. Second checkpoint is G2 checkpoint. This restriction point is located at the end of G2 phase. This checks the number of factors which are essential for cell division. Maturation promoting factor or mitosis promoting factor or Anaphase promoting factor, which is also known as MPF, is a protein composed of cyclin B and CDK1. This protein promotes the G2 phase into the entrance of M phase, and MPF is activated at the end of G2 by phosphatase CHK, which removes an inhibitory phosphate group added earlier. The main function of Anaphase promoting factor is in this restriction point are triggers the formation of mitotic spindle, promotes chromosome condensation, causes nuclear envelope breakdown. If there are any damages are noticed in this restriction point, then the phosphatase not activate the MPF, M-phase promoting factor, resulting in the arrest of cell cycle in G2 phase till the repair of damaged DNA. This prevents the transfer of defected DNA into the daughter cells. Third checkpoint is metaphase checkpoint. This occurs at the metaphase, means during the M phase. Anaphase promoting complex, which is APC, regulates these checkpoints. This is also called spindle checkpoint. This checks whether all the chromosomes are properly attached to the spindle fiber or not. This also governs the alignment of chromosomes and integrity of the spindle. If there are any mistakes, then it delays the cell division in entering into the anaphase from metaphase stage, means cell arrest in metaphase stage. So this is the sum up of this session, and this session is powered by digital version 2.0 Jayoti Vidyapeeth Women's University Jaipur. And if you have any query, then mention in the comment box. I will resolve it. Thank you.